Today I presented at the meeting Biobanking and the creation of cancer models for applied research on cancer. So the issue of having a biobank and having materials available, particularly for the process of cancer research from the identification of a biomarker to a clinical application, is the fact that in cancer research 90% of cancers are based on tissue, and therefore that is a limited resource. And so the main issue is having alternative material available for cancer research. And one of the best ways to do this is by using what they call PDX or xenograft models, and so that enriches for cancer cells and provides more tissue for the cancer research programs. In addition to that, these PDX or xenografts, which are mouse models, can be used as in vivo methods for testing drugs, and therefore they're sort of a preclinical approach to being able to do the applied research. So one of the main challenges of doing that is that you have to try and collect the tissue while it's fresh, because you need to be able to implant it directly into mice, or one of the newer techniques, which is the three-dimensional organoids, you need to be able to create these cell cultures from fresh tissue. And that requires you working directly with both surgery and pathology lab, because you're integrating directly in clinical diagnostic routine, because you have to be able to collect the tissue at the moment that it's received up in pathology for diagnostics. And this is a very important issue, also because it affects the quality of the tissue that you're going to collect, but it also affects the potential diagnostics of the patient's cancer. And so therefore it has to be the pathologist that is fully involved in this point of contact. So before we diverge into research, it has to be integrated into the diagnostics. And it's very important that we do this for a number of reasons, and particularly with regard to the fact that cancer is a very heterogeneous disease, and therefore it can change throughout the tissue collection or through the actual tissue. So it has to be done in a certain way and following a lot of protocols and standards in order to ensure that the quality of the sample, or in fact both from a morphological and a molecular standpoint, you're actually collecting the tissue that you want to investigate. And so that's why, again, it's very important to be part of the pathology integration in routine, but also you have to be able to follow these standards. And in fact, one of the issues that we were discussing today were the newly developed standards by ISO, which is the International Standards Organization, that are dedicated to biobanking in particular, but they also do refer to what are the clinical medical ISO standards to ensure that we're collecting things in the correct manner and not interfering with other processes. Yes, so biobanking has been around as a discipline for more than 20 years, and I think it's safe to say it started from the NIH in the United States. But about 10 years ago, in fact, Time magazine put it as one of the most important things to change the world in the next 10 years. Now, it hasn't quite achieved that, but it certainly has helped the improvement of research, because one of the main problems with research, and particularly cancer research, is that we can't reproduce some of the scientific results that we come out with the first time round. And the reason for that is because there isn't a consistent quality and a consistent identification or definition of the samples that we use. And so that's why biobanking has become a discipline, so that it can guarantee that research results can be reproduced and therefore applied in a clinical setting and help improve cancer care, which is what all of this conference is about.